Good afternoon. My name is Alice Lichtenstein, and it's my pleasure to introduce Dr. Paul Jacques, today's seminar speaker. Paul has a long and perhaps I should say a very long association with Tufts University, particularly the HNRCA. He joined the center in 1982 as a research associate in what was then called the Division of Biometry, Epidemiology, and Scientific Computing. In 1999, he became director of the Nutritional Epidemiology program, and finally, and 5 years later, he rose to the level of a senior scientist at the HNRCA and professor at the Friedman School. Throughout Paul's career, he's been very active in the center, the university, professional organization committees. I had the privilege of serving with him for many, many years on a Friedman committee, and together we trudged back and forth between the HNRCA and Friedman through snow, sleet, driving rain, and the heat of summer. Over the years, Paul has mentored an impressive number of students and postdocs, and I think what's most notable is how they themselves have gone on to have very productive, independent scientific careers. Dr. Jace has presented more than 100 invited academic talks on both national and international level. He has been consistently successful in garnering research support, notably but not exclusively related to B vitamins, which we'll be hearing about today, resulting in seminal contributions. And his work has involved numerous cohorts, but I think some of his best known work involves the Framingham Heart Study. His first public manuscript was published in 1981, and today Dr. Jacques has over 360 peer-reviewed articles and has well over 50 book chapters and reviews. So I know Paul has a lot to talk about today. So with no further ado, I will hand the virtual microphone over to him.
Thank you, Alice. Well, my title today refers to B vitamin status. I'm going to focus on vitamin B12, but part of that discussion will also involve folate. What I would like to show you today, based on the years of evidence that we've accumulated, is that vitamin B12 clearly supports a healthy brain aging, but unfortunately I cannot. What I hope to show you is why it does not and recommend ways to better study this relationship. The interest in insufficient B12 as a possible cause of cognitive decline and dementia stems largely from observations in the 1980s showing that neuropsychiatric abnormalities, including dementia, were common in B12 deficiency in the absence of anemia. This led to studies on B12 and cognitive function in generally healthy participants starting in the late 1990s. So we've accumulated more than 25 years of evidence on the role of insufficient B12 in neurocognitive decline, but there's still no consensus. The premise of my presentation today and the project for which I'll present results today is that the lack of consensus is due largely to flawed evidence. After proving what I believe to be important background information for understanding the limitations of the existing evidence, I will present a summary of the rigor of that evidence, and hopefully with that summary, I'll convince you that my premise is indeed true. So I'll start my presentation with a brief description of the biomarkers of B12 status because understanding the nature of these biomarkers is important to understanding some of the weaknesses in the existing literature. I will then discuss the impact of cognitive decline on diet and nutritional status because this is something that's doesn't appear to be considered in many studies of looking at B12 on cognitive decline. Next, I will describe a model of age-related neuropathy leading to cognitive decline and dementia. Again, understanding the neuropathy, and particularly the age at which pathological start to occur, is critical for understanding the limitations of the existing literature. As part of that discussion, I will touch on normal age-related cognitive changes as well. And as there is evidence that the relationship between B12 and cognitive function is also affected by folate status, and failure to account for folate status may be an important limitation of prior studies on B12 and age-related neuropathy. I'll also provide a snapshot of the evidence of that interaction. With this background, I'll review the rigor of the evidence relating B12 status to age-related neurocognitive impairment, and then I will provide recent results from a study relating B12 to cognitive decline, dementia risk, and volumetric brain changes that address prior limitations of the existing evidence. So let me start with a description of the biomarkers of B12 status. These include total circulating B12 or cobalamin, a component of total cobalamin called holotranscobalamin, and two functional biomarkers of B12 status, homocysteine and methylmalonic acid. So total circulating B12 is carried by one of two transport proteins, haptocorin and transcobalamin. But only the B12 bound to the transcobalamin, which is called holotranscobalamin or holo-TC, is metabolically active. The holo-TC carries only about 20 to 30% of the circulating B12, and the remaining is bound to haptocorin. When individuals are in a negative balance, the amount of B12 attached to holo-TC falls, but there is no notable decline in total B12 concentrations until holo-TC falls to very low levels. So the next biomarker I'll talk about is homocysteine. So B12 has two major metabolic roles, and one is the synthesis of methionine from homocysteine. This reaction, 5-methyl tetrahydrofolate, is a substrate for this reaction as well. As part of this one-carbon metabolic cycle, B12 plays a critical role in cellular metabolism, including methylation and mitochondrial metabolism. And through its interaction with folate in the cycle, B12 is critical for nucleic acid metabolism and DNA synthesis. If either B12 or folate are limited in this cycle, then the conversion of homocysteine to methionine will be impacted, resulting in elevated circulating homocysteine concentrations. So I want to show this slide to demonstrate that relationship. Here you can see the relation between homocysteine and inadequate circulating B12 and folate. And you can see that homocysteine is only elevated at the lowest levels of B12 and lowest levels of those with the lowest folate status. But since homocysteine is elevated in both B12 and folate, when these are when these are inadequate, it's elevated. So it's a sensitive marker to both B12 and folate, but it's not a specific marker for either of these. The next, the second role that B12 plays is the conversion of methylmalonyl-CoA to succinyl-CoA. And if B12 is inadequate for this reaction, it will result in a conversion of methylmalonyl-CoA to methylmalonic acid instead. So this results in elevated circulating methylmalonic acid levels. So methylmalonic acid is a sensitive but more specific biomarker for B12 deficiency. And so this slide, again, demonstrates the relationship between MMA and inadequate circulating B12s, as we see only elevated levels of methylmalonic acid at the lowest levels of B12. So this slide shows a model of B12 deficiency described by Victor Herbert in the mid-1980s. And it shows B12 deficiency as a gradually progressive disorder. So it starts as a negative vitamin balance, which is indicated by a low holo-TC, and this represents a depletion of circulating B12. That's followed by a biochemical insufficiency, as indicated by elevated homocysteine and methylmalonic acid concentrations, and slowed DNA synthesis. And finally, it's manifest clinically as frank deficiency in the form of megaloblastic anemia and neurocognitive disorders. So next, let me just raise the issue of cognitive decline in diet. Cognitive function is critical not only for remembering what we eat and reporting what we eat, but cognitive decline can also affect dietary intake. This creates a complex two-way relationship when we examine diet and pathological changes in cognitive function. So cognitive decline can affect appetite, can affect nutritional status. It can make diet-related tasks such as meal planning, grocery shopping, and cooking more challenging. And also depression, which can also affect dietary habits, may be a manifestation of early preclinical stages of dementia as well. So again, in considering the role of B12 in cognitive function, we have to look at the potential effects of cognitive function on B12. So next, I would like to use a model of age-related neuropathy to discuss the timing of the changes in cognitive decline relative to dementia incidence. And using this figure, if we start at the diagnosis of dementia and work backwards, we see that that's the time when typically when MCI would mild cognitive impairment would be diagnosed. Then if we go back 5 years prior to that, that's really when we start to see changes in cognitive function, so declines in cognitive function. And if we go back even further, we can see evidence of pathology in terms of either tau protein or amyloid beta in the brain 20 to 30 years prior to that. But we know that cognitive decline affects diet. So if we want to assess diet or nutritional status without the influence of cognitive decline, we need to do it at least 10 and preferably 15 years prior to the diagnosis of dementia. I just want to quickly note that not all cognitive changes that we experience with age are pathological. Some are part of normal aging, as described in this slide of 3 important cognitive domains. Memory is the most common concern for many of us as we age. There are 2 major types of memory: declarative or explicit memory, which relates to our conscious recollection of facts and events, and non-declarative memory or implicit memory, which is related to memory outside of our awareness. For example, tying your shoe or riding a bike. So declarative memory declines with normal aging, but non-declarative memory does not. Language is another complex cognitive domain, which remains stable or even it might improve a little with some exceptions. One exception is visual naming, which is the ability to see and name common objects. And this remains stable throughout much of adult life, but declines at older ages. The other exception is verbal fluency, which is the ability to retrieve information from memory, which declines with aging as well. Executive function is another critical complex domain, which can be described as the management system for our cognitive function. It involves the abilities that allow a person to engage in independent and appropriate goal-oriented behaviors, which might include planning and organizing, problem-solving, reasoning, and one's ability to control one's attention, behavior, thoughts, and emotions. Most executive function abilities decline with normal aging. So I just want to note that I'm not aware of any studies of B vitamins and cognitive decline that have actually distinguished normal aging from pathologic cognitive decline. However, whether pathological or normal, age-related declines can affect our recall of diet and our dietary habits at older ages. So next, I want to briefly discuss what has been referred to as the high folate, low vitamin B12 interaction. This interaction was first suspected as a cause of the observed relapse and exacerbation of neurological symptoms in pernicious anemia patients who were prescribed high oral doses of folic acid. The basis for this effect became moot when it was clear that B12 administration, not folic acid, was the proper approach for treating pernicious anemia. However, with the institution of mandatory folic acid fortification in the United States in 1998, evidence of this interaction has again appeared. As seen in this slide, the distribution of folate status in the U.S. population changed dramatically with fortification, with median folate levels increasing almost threefold. Prior to fortification, folate levels above 45 nanomole per liter were very rare, but now they are very common and typically referred to as elevated folate. So the effect of elevated folate status on the biomarkers of B12 is shown in this slide. If we first look at panel A in the upper left corner, we can see as expected that homocysteine levels are higher in individuals with inadequate B12 compared with individuals with adequate B12. But also among those with low B12 status, homocysteine levels are significantly higher in those with elevated folate compared to those with normal folate. The same phenomena can be seen for methylmalonic acid, for holo-TC, in panels B, C, and D, respectively. So why is the effect of elevated folate on B12 potentially important for research on B12 and cognitive function? Because it may affect the relationship between B12 status and cognitive function. In this slide, we can see that there is no change in MMSE scores over 8 years as folate levels increase. But among those, and this is among people with adequate B12, the fill circles, among those with inadequate B12, the open circles, we can see that as folate levels increase to very high levels, many mental scores dramatically drop. While the high folate, low B12 phenomenon is still poorly understood, it may be important to consider when examining the relationship between B12 and cognitive outcomes. With that background, I'd like to provide my assessment of the rigor of the existing studies. While the quantity of evidence relating B12 to healthy brain aging is large, the evidence is mostly of poor quality. The reasons are many, including older baseline age of participants, which older ages may be inappropriate for studies of primary prevention, as many older individuals will likely have experienced some cognitive decline. Another important issue is the risk of reverse causation bias, and this is due to the interrelationship between cognitive function and diet, which will affect all cross-sectional studies and also longitudinal studies with short follow-up times. Another issue is the use of insensitive or nonspecific markers of B12 status, such as homocysteine being nonspecific and B12 being insensitive. The failure to account for a possible effect of elevated folate on the relationship between B12 and neuropathological changes for the reasons that I just demonstrated. And another issue is the heterogeneity of the cognitive assessment methodologies across studies and weaknesses in cognitive assessment in many. While this is a really important issue, it's not one that I will address today. However, I will address the failure to account for baseline cognitive function in most of the longitudinal studies. So I just want to talk a little bit about the different study designs. And so given the large number of cross-sectional studies of B12 and cognitive function, and the inability to assess whether B12 intake or status is affecting cognition, or whether cognitive function is affecting B12 intake or status. So I won't summarize the cross-sectional studies. Also, because B12 intake does not correlate well with B12 status in older adults. As I will show you right here, you can see this is the slide I showed before relating B12 status to homocysteine, and you can see that elevated homocysteines at lower levels. Well, this is in the same population, same individuals, we see no relationship between B12 status and homocysteine levels at older ages. So because of that, I will also not summarize dietary studies of B12 and homocysteine. I will consider longitudinal observational studies that are based on B12 biomarkers among participants without dementia and randomized controlled trials, including B12 interventions, except for the RCTs where participants had MCI or dementia at baseline. These trials were obviously not designed for prevention, but to test treatments. I did not limit observational studies to participants who I knew were free of cognitive decline because the baseline cognitive status was not clearly characterized in most of these studies. So again, before considering the rigor of the longitudinal studies, I just wanted to again highlight the issues that I'm going to focus on. And these will include an inappropriate baseline ages, again, because of the potential for preventing development of neuropathology decreases and the risk of reverse causation increases with age as cognitive decline is more likely at older ages. Also, another issue that I'll focus on is insufficient follow-up time, as it limits the potential for any preventive benefit, and because of the increased risk of reverse causation again, particularly in studies of dementia, due to potential undetected existing pathology at baseline. Another issue is the use of the nonspecific, the homocysteine and insensitive B12 biomarkers of B12 status, and nonspecific vitamin interventions in RCTs. Most of the interventions included multiple B vitamins. I do this because it's not clear if we see an effect with a nonspecific marker that the effect is due to B12, and we may miss effects using insensitive markers. And finally, the last consideration is the failure to consider the potential effects of folate on the relationship between B12 status and cognitive decline or dementia incidence. But let me just note right now that I am unaware of any longitudinal studies that have assessed the potential effect of folate on these relationships. So based on a literature review of longitudinal studies of B12 biomarkers in cognitive decline and dementia from 1998 through October 18th of this year, I identified 13 observational studies and 13 randomized controlled trials of cognitive decline and 15 observational studies of B12 biomarkers in dementia. So let me start by summarizing the observational studies of cognitive decline and seen in this slide. So we had, of the 13 observational studies of cognitive decline, 6 included the B12 biomarkers homocysteine and B12. One of these studies also included HoloTC and MMA. 2 included only B12, and 5 included only homocysteine. Only 1 of 13 studies clearly indicated that baseline cognitive function was adjusted for in the analyses. Of the 8 studies that used B12 as a biomarker, 3 observed protective associations for cognitive decline or MCI incidence. Of the 11 studies that used homocysteine as a biomarker, 6 observed protective associations for cognitive decline or MCI incidence. In the 1 study that used MMA and HoloTC, both higher MMA and lower HoloTC were related to cognitive decline. So you can see that most of the evidence for an association here is based on homocysteine, which is not specific for B12 status, or circulating B12, which is not sensitive to B12 status. So this slide shows the distribution of these studies by baseline age and length of follow-up. So the lowest mean age of participants in all 13 studies was 62 years. The mean baseline age was 75 years or older in more than half of these studies. And the average follow-up time was 5 years or less in all but one of these studies. For dementia, we had 15 observational studies. 2 of these included biomarkers for homocysteine and B12. 1 included B12 only biomarker, and 2 included homocysteine, holo-TC, and 10 included only homocysteine. Only 1 of the 15 studies indicated that participants with cognitive impairment were excluded, and 2 indicated that baseline cognitive function was adjusted for in analyses. Of the 3 studies that used B12 as a biomarker, 1 observed a protective association for dementia. Of the 2 studies that used holo-TC as a biomarker, 1 observed a protective association for dementia. And of 14 studies that used homocysteine as a biomarker, 10 observed protective associations for dementia. And again, most of this evidence for association was based on homocysteine, which is not specific for B12 status. And again, this slide summarizes the follow-up time and baseline age for the studies of B12 and dementia. And the lowest mean baseline age of all participants in all 15 studies was 47 years. Second lowest was 63. The mean baseline age was 75 years or older in 53% of these studies. And follow-up times was 5 years or less in 2/3 of these studies. Now, given the timeline I showed earlier regarding the timing between MCI diagnosis and dementia diagnosis, the risk of reverse causation would be very high for most of these studies, given that the vast majority of dementia cases would have moderate to advanced MCI at baseline. And incredibly, the one study that reported excluding cognitive impaired participants at baseline only had a 4-year follow-up. I'm not sure how that happened, but there was one exception here, and that was the Gothenburg study of women. This is a figure from that study. This is a study that included the youngest participants, mean age 47 at baseline. And I think it's very informative regarding the risk of reverse causation bias. I want you to note that the divergence in the rate of dementia does not occur until almost 20 years after baseline. You see that right about here. In all of the other studies of B12 status and dementia, the divergence would have had to occur within a few years after baseline. And in the 2/3 of the studies with 5-year follow-up or less would have to have occurred within 1 to 2 years of baseline. This strongly suggests that reverse causation bias affected most of these dementia studies. This slide summarizes the evidence from the randomized controlled trials. There are 13 trials of B12 supplementation and cognitive decline performed in subjects who were not cognitively impaired at baseline. At least they didn't indicate that any of them were cognitively impaired. 10 trials used a B vitamin combination that included B12. 3 used B12 as a tablet or injection. None of the 10 trials that intervened with B12 or the B12 combination showed any cognitive benefit. Of the 3 trials that intervened with the B12 intervention, 1 showed benefit. Among these 13 trials, the lowest mean baseline age of participants was 60 years. The mean baseline age was 75 years or older in 55% of these studies, and the average follow-up time was less than 2 years in 2/3 of these studies. Let me end the review of the longitudinal studies here with studies of B12 status and neuropathy based on those studies of brain imaging. There are no longitudinal observational studies of brain imaging. There were two similar trials that intervened with B12, B6, and folate. These were combined into one retrospective pooled analysis, which is presented in this slide, and it shows that the B vitamin intervention significantly reduced the rate of whole brain atrophy over a 2-year period. Again, this intervention was not specific to B12. So just let me summarize basically the rigor of these studies. The studies with the older baseline ages, whether observational or trials, limit the quality of evidence, as most participants, whether due to pathologic or normal aging, are more likely to have loss of cognitive function, and many are beyond the age where interventions may be most beneficial for preventing neuropathology or for maintaining cognitive function into older ages. The risk of reverse causation bias, which is very high in these observational studies of older participants and may account for beneficial associations between B12 and neuropathy. And this is amplified. This concern is amplified because most of these studies did not exclude individuals with impaired cognitive function or adjust for baseline cognitive function. Short follow-up time also increased the risk of reverse causation bias in observational studies, particularly in studies of dementia, where many participants may have had advanced MCI or as yet undiagnosed dementia at baseline. The evidence is almost entirely based on nonspecific or insensitive biomarkers of low B12 status in observational studies and nonspecific vitamin interventions and RCTs. Moreover, many of the trial participants were selected based on elevated homocysteine MMA and low B12. So it's possible that the limited protective benefits seen in these trials may be due to improved cognition in individuals with insufficient B12 status and not due to protection against cognitive decline. And finally, as I previously noted, none of these studies considered the potential effects of folate on these associations. So with that background, I would like to present some recent published findings and some work in progress from our study of B12 status in brain aging. The primary objective of this project was to examine the relationship between B12 status and cognitive decline, dementia incidence, and changes in cerebral volumes while addressing limitations of the prior longitudinal studies. And these include using a more sensitive and specific B12 status biomarker, using a large, well-established, long-running cohort of middle-aged adults with 3 assessments of B12 biomarkers across approximately 15 years, and examining if any relationships between B12 and status or neuropathological outcomes are affected by folate status. So the findings I'll present were done using data and blood samples from the Framingham study offspring cohort, and these were collected between 1998 and 2014. Today I'll present findings from our recently published article examining the relationship between B12 status and cognitive decline, and from the preliminary analyses on dementia incidence and changes in hippocampal volume. Our cognitive assessment was based on a neuropsych test battery that included a variety of tests, which are listed here. But the primary outcomes were change in memory, executive function, and language, and these were based on cognitive domain factor scores. In the Framingham cohorts, dementia is assessed by a continuous surveillance of study participants to identify possible dementia cases. Oops. if surveillance suggests a decline in cognitive function, a consensus review is conducted to determine if these participants meet the criteria for a diagnosis of dementia or Alzheimer's disease. Brain MRI scans were performed up to 3 times to characterize changes in brain structure, total brain volume, lobe, gray and white matter, hippocampal and white matter hyperintensity volumes were assessed, as well as cortical thickness. And these have been assessed longitudinally. Today, I will present data on a small subset of participants for whom we currently have only 2 MRIs approximately 7 years apart. And to address the concerns with the use of insensitive and non-specific markers of B12 status, we used a marker of B12 status that mathematically combines B12, holo-TC, methylmalonic acid, and homocysteine in their continuous forms while adjusting homocysteine values for folate status. And this is referred to as the CB12 score. This marker presents an advantage as a combined assessment of B12 status for all stages of B12 inadequacy. For this project, we applied a validated 3CB12 version based on B12, methylmalonic acid, and homocysteine because we did not have sufficient sample volume to measure holo-TC. And as you can see, based on the categories of the 3CB12 marker, over 98% of participants in our sample have adequate B12 status. This is a fact that may be important as I present some of our findings. So this slide describes the characteristics of a sample. These data represent a sample from our recently published paper, but the dementia sample is very similar in characteristics to this. The mean age of baseline is just under 60 years old. Over 40% are college graduates. 22% have the ApoE epsilon 4 allele. They're on average, they're overweight. And about 42% have elevated folate, which is pretty typical for Americans. And the follow-up time was 14.2 years. So this slide shows the association between the mean annual cognitive decline in the upper 3 quartile categories of cumulative average 3B12, 3CB12, relative to the lowest quartile category. A mean positive difference indicates that the annual cognitive decline was less than observed in the lowest C3B12 category. While all factor scores declined over the 14-year follow-up, you can see that the higher B12 status was significantly associated with a slower decline for all cognitive domain factor scores. For each factor score, the difference in annualized change was significantly less in the highest versus the lowest category. So we also examined the potential interaction between folate status and B12 status with respect to cognitive decline. But as shown in this slide, there was no evidence was apparent. You can see that except for the 3rd quartile for the memory score, for the non-elevated represented in the lighter bluish green versus the elevated in the darker blue. There's no significant difference between those with the high and low elevated and non-elevated folic acid for these the 3 factor domain scores. So our sample, there's 2 explanations for this. It might be that higher folate does not affect the association between B12 and cognitive decline. Another explanation might be that our sample did not include enough individuals with low B12 status to detect this effect. As you recall, 98% of the people in this sample had adequate B12 levels. So this slide shows some preliminary data on the relationship between the 3CB12 score and incident dementia, basically showing the relationship quartiles, the upper three quartiles of the C 3CB12 score relative to the lowest quartile category. The relative risk relative to the lowest quartile category. And it's stratified based on the elevated and non-elevated folate. And as you can see, there's little evidence of any overall association between the B12 status marker and dementia risk, and little evidence of any difference in the association based on folate status. As I mentioned, these findings are preliminary. We just received these the data for these to do these analysis last week. So I anticipate some potential changes to the inclusion criteria as we investigate this relationship in more depth, but I don't anticipate that there'll be a great, the change will greatly impact our findings. So finally, let me present some, again, very early findings relating the 3CB12 score to our neuroimaging data. These analyses were performed on a small subset of 297 Framingham cohort members with at least 2 MRI scans. We examined the relationship between the B12 status and decline in hippocampal volume. A positive slope, again here, represents a slower decline in hippocampal volume as the 3CB12 score increases. We detected a significant age interaction for these analyses. As you can see, there was no indication of any association for those with baseline ages less than 60, which you might anticipate based on the lack of large volumetric changes in the brain in younger people. But we did see a slower decline with higher 3CB12 scores for those who are greater than or equal to 60 years old. So we're currently updating, getting updated or awaiting updated data from our colleagues at Framingham and MGH on the neuroimaging, which will include over 1,000 participants with 2 or more scans. So in summary, I hope I've convinced you that the existing evidence for a benefit of higher B12 status for prevention of cognitive decline and dementia is equivocal and largely of low quality. Future studies need to account for the complex relationship between diet and cognitive function in study design and analyses. There's only one longitudinal study of the relationship between B12 status and neuroimaging with one trial. There's no studies of which I am aware that examine B12 status in any of the newer circulating biomarkers of brain damage and Alzheimer's disease. Oops, sorry. Our recently published work relating B12 status to cognitive decline based on an improved approach included younger age, longer follow-up, accounting for baseline cognitive function, use of sensitive biomarker, B12 biomarker, and consideration of the potential folate interaction. And with this, we've demonstrated that those with a higher B12 status have a lower cognitive decline. Our preliminary analysis on brain changes support that relationship, but our preliminary analysis on dementia incidence do not. It could be possible that the cognitive decline association that we're seeing is based on benefits to normal cognitive aging, not pathologic, and therefore no association with dementia, but that is inconsistent with the neuroimaging result. And finally, our findings do not provide support for an interaction between B12 and folate in relation to cognitive decline. And so I would like to end by acknowledging all of those responsible for this new research that I've presented here today. Josh Miller, my co-PI on this project from Rutgers, Philip Wong and Francesca Marinaro from BU. I want to thank them for their wonderful work on the cognitive decline paper. Doug Grieve, my colleague on neuroimaging at MGH. Jacques, a consultant and longtime collaborator on this project. Josh Rutt from the HNRCA and Gail Rogers from BU and the Framingham study for their invaluable aid with the Framingham data and statistical support. And my longtime collaborators, Joel Mason, Irv Rosenberg, and Rhoda Au, who are indirectly responsible for this project through their support and mentoring over the years, which has made this project possible. And thank you.
Thank you. Thank you, Paul. That was quite a provocative presentation. I would like to remind the audience that if you have any questions, please put them in the Q&A and I will read them out. Waiting for those to come in, you've given us a lot to think about. And we're all aging. Everyone on this listening to you now is aging and who will listen to you in the future. So I guess my bottom line question to you is, what's the best thing on the basis of all that you've talked about? What's the best advice you can give people in terms of healthy aging and something I think we all want, to maintain our cognitive status?
Yeah. Well, you know, I think that, I mean, again, it goes back to the evidence. Even, you know, you look, there's a lot of evidence just healthy diet patterns. And there's good reason to believe that healthy diets, including B12, would be beneficial for preventing cognitive decline and for lowering the risk of dementia. But again, the data supporting them is relatively weak too, for many of those reasons that I've discussed today. But that said, I think that there is sufficient evidence that we should maintain a healthy diet, a healthy, more plant-focused diet, I think is the best. And you know, I think making sure, particularly for older individuals, I think a B12 supplement is probably not a bad thing to take.
Well, particularly since you mentioned plant-based diets, and plant-based diets are going to be the diets that are lower in B12. Well, we do have some questions coming in. So from Jiandong Wang, it is known that patients with atrophic gastritis or autoimmune gastritis or undergoing metformin therapy display reduced vitamin B12 status. I'm wondering if there's any data to show that patients with atrophic or autoimmune gastritis could increase cognitive decline, or supplemental B12 for those patients could prevent the decline, I guess, preventing.
Yeah, I mean, we know that these individuals have low B12 status, and so it is a concern. And again, we know B12 deficiency, which many of these patients may be not very far from, in terms of that continuum, is associated with cognitive decline because of issues, many issues in terms of the B12 functions. But so yeah, I mean, I don't think there's any real large studies that have looked at these individuals specifically that I'm aware of to see if there's any impact on cognitive decline. But there is an impact on B12, and we have to. I mean, we might not know whether B12 status in individuals with adequate B12 affects cognitive decline, but we know that B12 deficiency affects cognitive decline. So yeah, I mean, it's something to be concerned about.
Okay, from Joel Mason, Paul, in regard to the present study, were the folate levels you described total serum folate or serum folic acid levels? And would you like to speculate as to if there might be some relationship if it was the latter were examined?
Okay, yeah, and I'll be happy to answer that. I mean, these are serum folate levels. If you really can't see, when you look at folic acid levels in the serum, they tend to be quite small because you're in a fasting state. But you can't get folate levels over 20 or over basically 43 nanomole per liter or 20 nanogram per milliliter, you can't get those without taking folic acid. It's just almost impossible. I mean, I imagine you could get clear to that, but it'd be impossible. So anyone with elevated folate is taking large quantities of folic acid.
Okay, thank you. Then Sarah Booth said, Paul, a wonderful review on this important topic. How do you envision the message of B12 and cognition for the general public through healthcare providers based on homocysteine levels, or just recommend over-the-counter dietary supplements?
Well, it's a good question because homocysteine is something else. I mean, you know, there is evidence that homocysteine itself is a risk factor for cognitive decline and dementia, independent, not necessarily independent of B12. But we can't in those homocysteine studies, we can't really identify B12. I mean, the Gothenburg study, for example, that I presented, that was based on homocysteine and showed that over many years that the risk of dementia was higher for those with elevated homocysteine. So I don't think it can really separate homocysteine too much from B12, although it might not be B12 specifically that's causing the dementia in that case. So it's a good question. So should you have your homocysteine levels tested? You know, for older individuals, that might not be a bad idea. But for most of us, I think that just taking a dietary supplement would be sufficient. A dietary B12 supplement.
Are you suggesting B12 alone or B12 as part of a multivitamin?
Well,
To put you on the spot.
yeah, no, maybe B12. And the problem is multivitamins all contain folic acid. So you're increasing your folic acid. I mean, again, we don't know if really the nature of this high folate, low B12 interaction. I suspect it's unlikely that people who are taking a multivitamin would have sufficiently low B12s to be impacted, unless they're having absorption issues with the synthetic B12. But so I don't. It's kind of a hard question to answer. But I think, I mean, I'm not a supplement pusher necessarily, but I think for older individuals, a little extra B12 probably isn't going to hurt.
Yeah. From Chris Wiley, I love your perspective, the perspective you offer. Out of curiosity and as a mechanism-focused researcher, do the differences in these biomarkers as readouts hold any implications for which pathway a vitamin B12 might be acting on, he's saying, the improved cognitive function? I assume sort of the slowing decline, I assume.
Yeah, slowing. Yeah, it's hard. I mean, because B12 could be involved in so many different aspects. It's critical for mitochondrial function. It's critical for myelin synthesis and repair. It's critical for a number of neurotransmitters. Some people believe it's associated with inflammation, and lower levels are associated with higher inflammation in the brain. So, I mean, there's a lot of B12 is so essential to so many things, including DNA synthesis. That it's hard to pinpoint a mech. I mean, I would want to, go out on a limb and say I think it's this or I think it's that. I think it's probably a lot of things together that could be adding up.
Okay, and from Irv Rosenberg, what do these same studies show if homocysteine is the putative causal agent with cognitive decline instead of B12 alone?
The same thing, because they still all have the same, for the most of them. I mean, again, as I mentioned, that Gothenburg study, I think was a reasonably done study starting in younger ages. But when most of the participants you're enrolling in your studies are already seniors, already have some level of normal cognitive aging. Um, it's if you're looking after you look at the pathology, if you want to prevent the pathology, you have to go back even farther. You have to go back not 15 years prior to the development of dementia, but you have to go back another 10 to 15 to 20 years if you really want to slow the pathology. So at least some of the biomarkers of pathology. So it's the homocysteine. I mean, still the homocysteine studies are they're still done in older individuals. I mean, that takes away one of the concerns. If homocysteine is a neurotoxin, then the lack of specificity isn't a problem when you're doing a lot of these trials and a lot of these studies. So it takes out one of the issues that we have with B12, but it doesn't remove the other concerns.
Okay, thank you. If somebody else has a question, they should get it in very quickly. Otherwise, I would like to thank you. It really was such a thought-provoking presentation that I suspect a lot of us will be thinking about it for a while, and you may get some questions afterwards. But since no other questions have come in, once again, thank the audience for listening and participating. And Dr. Jacques, thank you very much for your presentation.
My pleasure.